Um, I hope you are healthy. Please tell me the number of slides I must teach today. And please tell me the number of slides, please. I think we have finished the omics techniques, aren't we? I think we have finished it. I don't know if we must teach the omics techniques right now. Did you read this table? Did I tell you the advantage and limitation of the omics technology? Did you read this table? Please answer me. The advantage and limitations of the omics technology. Did we finish the omics technology? Metagenomics, protogenomics, transcriptomics. And did you read the protogenomics? Left formation on a taxon, the morphology can include the colony characteristics, color, shape, pigmentation, production of a slime, etc. Further, the features of the cell are described as to shape, size, gram reaction, extracellular material like capsule presence of endospore, flagella presence, and location, motility, and inclusion bodies. Like you describe the board cell features. So the first phenotypic method is characterization of the morphological traits, such as colony, such as they are mobile or not, and we can investigate these traits uh, on light microscopy. Okay, Mr. Basil Hari. Okay, thank you. So we must start the power number 3.
Okay, thank you. Did you tell the other students to come, please?
To review the topic for the last section that you have read it, numerical taxonomy, cellular composition, and fatty acid analysis we teach. Yes. And isoprenoid quinones and other diagnostic methods, advanced spectroscopy and spectrometric methods. And now today we are going to start the genotyping methods, yes? Okay. Genotyping techniques. Modern taxonomy has been influenced by genetic methods, and indeed much of the classification and identification is predicted on specific gene sequences. All the techniques involving DNA or RNA fall under genotyping methods. So the modern taxonomy that I told you is the polyphasic taxonomy is concerned on two techniques together, phenotypic techniques and genotypic techniques. Today we are going to study the genotypic techniques, and the first of all is 16S rDNA based analysis. The technique, which is very nearly a gold standard for taxonomic purpose today, is sequencing of the 16S RNA gene of bacterial multiple alignment programs such as class X, class W, class X2, class W2, mega, tea coffee, muscle can also be used with manual editing. So you can see many programs that can be used in editing of the program sequence that we name here. Class X, class W, class X2, and class W2 are good. The mega program is more common, and tea, coffee, and muscle is important. Can also be used with manual editing. Subsequent to alignment, phylogenetic trees or dendrograms have to be constructed to reveal the taxonomic position of an organism. So when we sequence the 16S RNA gene, we have to draw a dendrogram of phylogenetic tree to investigate the relationship between our bacteria, our favorite bacteria, or the bacteria that we have isolated with another bacteria or another species, another genus, and another taxon. So drawing the dendrograms or phylogenetic tree is needed to investigate the relationship between species. And here you can see the picture for these data sequencing. You can see here we have sequence S. I'm going to get the figure for you. Here you can see studied rRNA or 16 rRNA. This is our sequence X, for example, sequence X that we have isolated from environment. Okay, here we have the sequence database. First of all, we performed PCR and then sequence with sequencer. And here we have the sequence X. Okay, then we use the BLAST program. We put the sequence on this programming computer, and then this computer with Fluxal Omega Map can, with this program, can tell us the similarity and the probably the species that our sequence X is contained. You can see here the number of Clavibacter, and you can see the list of bacteria that are proposed that our sequence X is that. Depend on their similarity. Okay, and here I told you this is the continuity of the picture. Fluxal Omega Map can tell us the similarity of sequence of our sequence, I mean sequence S with another genus sequence, the sequence that is similar. For example, for Leucobacter, for Cryobacterium, Microcella, and here is sequence X for us. Bacillus, Acinetobacteria, Frigoribacteria, Acinetobacterium, sequence X. Clavibacter is very near. You can see here, Clavibacter is very near, so it can be a species in Clavibacter genus, Cortobacterium. So figure 6.8, key steps in phylogenetic analysis. You know, what's the meaning of phylogenetic? Phylogenetic means relationship, a near relationship between bacteria. Okay, A similarity, search for sequences, homologous, sequence of interest. The sequence of interest is sequence X. Our interest for sequence X. In the sequence database, extraction of compartments from causal related species in B. So the Fluxal Omega Map program can tell us the similarity sequences near sequence X or sequence of our interest. So here you can see many genus that are near to our genus respect to their sequences. Okay, and you can see the causal related species here and multiple alignment of sequences. And for the next step, D, filtering the alignment, you can see here. From here, we was here, region where homology between site is reliable remain, and you can see the DNA list here, construction of a distance, magnification of closest known relative. Okay, so we can use these programs for drawing the dendrogram. You can see here the dendrogram or phylogenetic tree. Okay, so we have here the phylogenetic tree. As you can see, this is our sequence of interest, or sequence S. Proteobacterium is the nearest genus to the sequence X, and then after that Clavibacter, and then Bacillus, Acintobacterium, Figobacterium, Leucobacter, and the forest genus here is Acintobacteria. So the dendrogram or the phylogenetic tree is drawn based on relationship between bacteria. If the bacterium is far from our sequence, is has less relationship with our bacterial sequence. So the distance here is very important. If the distance is too far, so they are far from them. And if the distance is near, for example, Proteobacterium, these genus are very related to each other, and we can see the near relationship between them. So the distance in this dendrogram can show us the nearest and the farthest genus respect to their phylogenetic similarities. Okay, if you have any questions from these pictures, please ask me. Okay, and the second genotyping method is DNA-based content. The first was 16S rRNA, and the second is DNA-based content. Here you can see a determination of mole percent guanosine and cytosine constituents, a classical method of establishment of genomic content. So the DNA-based content means guanine plus cytosine content. So determination of percent of these moles is very important, guanosine plus cytosine. And we can see the similarity of this percentage in different bacteria and also in different organisms. For example, we want to see the similarity between elephant and monkey. For example, we can measure the percent mole of combination of guanine plus cytosine, and then we can comprise to each other. And then if they have similar percentage of guanine plus cytosine, they are related to each other and they have phylogenetic relationship. This is now being used along with other genotyping methods to establish taxonomic position of an organism. Within a species, the guanine plus cytosine content ranges within 3% and within general 10%. Overall, the guanine plus cytosine content ranges from 24 up to 76% in bacteria. So, which in general 3% and up to 10% we can see, and in bacteria, that is very important, 24 up to 76% in bacteria. We can see the percentage of this content, I mean guanine plus cytosine mole percentage. So another way is calculating of this percentage. You can see here figure number 6, composition range in DNA-based guanine plus cytosine percentage. You can see here the formulation of this calculation, okay? Guanosine plus cytosine percentage mole. You can see at the top guanine plus cytosine mole, and you can see at the bottom mole guanine plus cytosine and plus adenine plus thymine. And this is the formulation of calculating of this percentage. And now we can see here composition range in DNA base. You can see here prokaryotes, we taught that 24 up to 76. Okay, do you remember the previous power? Here we taught 24 up to 76. You can see here. Okay, so the range here is 24 up to 76 in bacteria, and in archaea is similar to. In the eukaryotes, this percentage is less. Beauty planets, photosynthetic, unicellular, fungi, and protozoa, this content is accurate. So you can see the range of DNA bases in prokaryotes is more than eukaryotes. In animals, eukaryotes, and also in eukaryotes microorganisms such as fungi and such as protozoa. So the prokaryotes are at the top for the most range composition of DNA bases. And the third technique is DNA hybridization in genotyping techniques. DNA plus DNA hybridization. This method is an indirect measurement of sequence similarity between genomes. A cutoff value of 70% similarity is considered for establishment of species. Hence, it is applied only where 16S rRNA gene sequences show similarity value above 98%. There have been reports where 16S rRNA okay. Okay, so here you can see another technique, DNA hybridization, and in this technique we must measure the similarity between sequences and between genomes. How we can do that? first of all, we have to denature the two strands of DNA. You know, the DNA is double strand. So, first of all, we have to separate them from each other. So, we must do hybridization between single strand DNA and another single strand from the bacteria that are the same as our bacteria. For example, we isolate environments and we guess by biochemical tests, morphological characterization, we distinguish this bacteria that we have isolated is, for example, Bacillus cereus. Okay, so we are going to isolate the DNA and then we must denature the DNA. We must separate the two strands from each other. So one strand from our interest, Bacillus cereus, for isolate that I have picked it up from environment, and another strand from Bacillus cereus that is communicable and uh, informal communicable. They are exit. So uh, unknown bacteria, we get Bacillus cereus. Unknown bacteria must be Bacillus cereus. Then we must do hybridization between these two strands. If the percentage of hybridization is 100, so that means the bacteria are the same and our distinguishing is correct. But if the percentage is less than 100, we must see the similarity between them respect to species or genus. So hence it is applied only where 16S rRNA gene sequence shows similarity value about 98%. They have reports, and when the similarity of DNA hybridization is more than 70%, that means they are the same species. Okay, and this, please note, this was a question for final exam, the previous term. The percentage of the hybridization here upper than 70% is equal to 98% similarity in 16S RNA gene sequencing. So in this technique, the similarity must be upper than 98% for diagnosing in the same species. But in this method, the similarity must be upper than 70%. So you can see the percentage in this technique, the DNA hybridization, is different from 16S rRNA. Here must be upper than 98%, upper than 98% is equal to upper than 70% similarity between species. So we can say they have the same species, not the different species. Okay. If you have any question, I give you 1 or 2 minutes to continue. This is the picture of DNA hybridization, and I'm going to tell you one more time. As you see in this picture, number 6.4, method of DNA hybridization, schematic and principle of the method. The DNA hybridization is done between the DNA of two organisms. As I told you, one organism is known and another is unknown. Here is numerated with 1 and 2. The DNA of one organism is labeled with radioactive phosphor 32, and the DNA is then denatured, double-stranded hybrids between microorganisms, and removal of remaining single strands by enzymatic digestion. Okay, as you see here, first of all, we have DNA preparation. DNA preparation means um, conversion of two strands to single strand. This is organism number 1, that is known organism. DNA extraction, we have for organism 1 and 2. Fragmentation and labeling with phosphor 32, you can see here. We have double strand. Here we first of all label this, then denaturation is done for organism number 1 and 2, and then we have here a single strand, we can do the hybridization. Okay, here it's shown hybridization between organism 1 and organism 2. You can see here the hybridization between single strands. If the percentage of hybridization is 100, its two organisms are the same, and we can take and we can use the known microorganism as control for 100 of hybridization, 100% of hybridization. Okay. Other genotyping methods. Earlier, subtyping was done on the basis of biochemical profile, biotyping, serological profile, or serotyping, phase susceptibility, or fudge typing, or antibiotic susceptibility. So all of these techniques are genotyping method too. When we investigate about biochemical properties, we are doing biotyping. When we are searching for serological profile, we are doing serotyping, and fudge susceptibility is named fudge typing or antibiotic susceptibility. But currently, DNA typing methods are preferred due to their reproducibility, ease of performance, and high level of discrimination between strains. Genotyping methods such as restriction fragment length polymorphism or RFLP, randomly amplified polymorphic DNA rat. You have read it before. RFLP or fragment length polymorphism we have studied, and randomly amplified polymorphic DNA or RAFT we have studied too. So these are another genotyping methods too. Amplified A for amplified, F for fragment, L for length, and P for polymorphism. Amplified ribosomal DNA restriction analysis. You have studied it before. Do you remember it? ARDRA. The technique ARDRA, repetitive element polymerase chain reaction, or REP-PCR. That is a kind of PCR2. Ribotyping and multi-locus sequence analysis, or MLSA, are some of the newer methods to characterize a taxon. So when we want to characterize a taxon, we can use these genotyping methods for this distinguish the genus. And the next title is Phylogenetic Tree. Okay, because your teaching is not behind, I told you before, we stop the teaching until this title, and I will teach the next section for you. Phylogenetic tree. Please write number of the slide is 42. Sorry, 24. Slide 24. And the number of power is 3. So the next section we are going to teaching the phylogenetic tree, the programs of this method, and the explanation of that. If you have any question, you can ask, and if you don't have any question, I will say goodbye until the next week.